Hello everyone, and welcome to today's webinar, Improving Protein Expression in E. coli B strains using Rapid Genome Engineering. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, please visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Tyson Shepherd, PhD, Microbial Engineering Applications Manager at Inscripta. Dr. Shepherd, you may now begin your presentation.
Good morning. Thank you so much for joining us for this morning's webinar. So I'm a senior scientist and applications manager at Inscripta. And today I'm going to be talking to you about applying the Inscripta platform technology with its capabilities of rapid genome engineering to improve the expression of E. coli B strains. So as you probably know that the E. coli B strains are a go-to resource for the heterologous expression of proteins. And here we thought, wouldn't it be interesting if we could apply the genome engineering capacities, the rapid and high throughput capacities of the Onyx platform to be able to investigate variants of those B strains that could improve production of proteins. And so that's the story that I'm going to tell you about today. So as you are all probably aware, heterologous protein production is forming sort of the cornerstone of the entire building of a bio-based economy. And we see this happening throughout a number of different areas within society that are being completely transformed over the modern era with the capabilities of that come along for DNA synthesis and rapid DNA and cost-effective DNA sequencing. So this has been a long time coming, but we're seeing brand new capacities being built within industrial biotechnology for improving fuel production in a green way, being able to degrade waste streams and create valuable commodity chemicals from those waste streams and generate new dyes and new concretes and all sorts of fantastic things that are coming from industrial biotechnology. When we look at other areas, you're probably aware of the alternative protein productions that come from synthesis to make what can be called lab-grown meats or proteins that would synthesize the experience of eating meats in that flavor profile and nutritional quality. The other another fields of interest is the impact of synthetic biology on agriculture. So that comes in the form of, you can think about nitrogen fixation for certain plants, as well as being able to engineer better microbiome for improved crop yields or generation of insecticidal proteins. There's a number of very cool applications that are being built within agriculture. And then, of course, in the final one I have listed here is in biopharma and therapeutic production. And that comes from the fact you can think of in living medicines, the capabilities of having at the site generation of therapeutics, and then also therapeutics being produced within E. coli itself or yeast or human cells. And those span the gamut from enzyme replacement therapy through generation of specific antibodies. So all of this is being built on the aspect that we can do rapid engineering of host cells, and these microbial cell factories are that production element that's necessary for advancing all of these industries. But when we think about the problem of heterologous protein expression, there's usually a few challenges along the way. When we set out for improved protein production, what we're realistically looking for at the end of the day is improved yields of functional enzymes or proteins and improved purity of those proteins. And there's a number of headwinds that are facing us when we look at that yield and purity profile for getting improvements. And those might come from low expression of the protein of interest. The protein may be have solubility issues or problems with forming inclusion bodies that would precipitate out of solution and be inaccessible for subsequent refolding, let's say. Certainly degradation is an issue with the presence of proteases, especially upon lysis in E. coli. And then we think about the toxicity profile frequently that comes with E. coli, that it has endotoxins that have to be removed that can be costly for that removal. Proteins can also become misfolded if not, if it requires a specialized chaperone or growing conditions. And then, of course, the ultimate form is the active protein of interest or active enzyme, and we may have inactivity due to any number of other problems. So there's a number of, all of these headwinds come about and getting us to think, well, how do you overcome those as the state of the art? And ultimately what it comes down to, and what I'm going to propose to you, is that it's all, for the most part, all of these can be overcome by scalable host strain engineering to meet the demands of the protein that's being produced. So that host strain engineering means what type of modifications can you do to the chassis or the E. coli or the yeast to be able to improve the expression of an otherwise a protein that would not be happy being produced in its current circumstance. So when we think through the actual production of the protein, usually it's comes down to a number or a thought process that the individual takes when generating the final protein. So that can be in the form of how to create the actual construct itself with the promoters, the gene itself, and that optimization of the coding sequence, which terminators to use. What does the structure look like? Are there any modifications that you might want to build into it? Should I be integrating this into the genome, or should I be producing it off of plasmids? These are the type of thought processes that go into the specific target gene of interest that you're looking at. And then there's the sort of the next step of thinking about is, what host cell do I want to use? So as I mentioned, a go-to host cell within the community is, of course, like B strains, like BL21, for example. That might be your first go-to cell. You might want to express it due to chaperones or due to translation, due to glycosylation. You may prefer yeast, for example, or a variant thereof, or you may need to go all the way to human cells in order to allow for that. There's a number of different varieties of host cells that I'm going to talk about in a few minutes. And so that's not an inconsequential decision when you think through what your host cell looks like. And then at the end, you grow up your host cell in some type of media. So you're thinking about the media, and you're thinking in terms of how to get that purification. So that might be, you might want to introduce fusion tags into it. You're probably looking for a very specific amount of the protein of interest, looking specifically at what confounding factors are there, such as endotoxin levels. Can you secrete the protein rather than having it internal to the cell and avoid lysis, etc. And then finally, you have your purified protein. That's where oftentimes a lot of us in the audience think that that's you've reached something exciting. Then once you have the purified protein, you can test its activity and its capabilities. And so that's what we want to get to is as quickly as possible, ultimately, is that purified protein that we can take downstream for structure activity relationships or functional activities and new discoveries. So we want to make that an easier process, because right now the state of the art is there's a number of different strains. When you think of, like, for B strain production of proteins in BL21s, for example, and derivatives, these are often the strains that you go to for protein production. And there's some very good reasons for going to these strains. So BL21 has a LON and ompT proteases. It's deficient in these proteases. So you're going to have improved yields and less degradation. B strains themselves have an improved ATP utilization, possibly or principally due to the fact that they're non-motile, and so that ATP resource can be diverted to greater protein production. It also has reduced acetate during protein production. So there's a number of reasons for BL21s being valued for protein production over K strains. So that's the alternative. That's what we've talked a great deal about that in previous webinars with utilization of K strains, where you're thinking more in terms of cloning and genomic discovery. Here it's more about Hardin's protein production is a go-to resource for these. So when you're thinking about basic protein production, there's the BL21DE3s. If it's a toxic protein, you might want the P-lyse in there. There's PET plasmids as your go-to resource for production, typically with the T7 polymerase on that DE3 lysogen that allows for production off a T7 promoter. And then you do some moderate condition screening with different media types, as well as different temperature of induction, different flasks, and so forth. These are kind of the basic or first rounds details on looking at that. But often we'll want expression of proteins that, say, have a human codon bias associated with them. E. coli is not going to like that. So we might want to diversify our strains a little bit. So we go back to our freezer, we might pull out the Rosettas or BL21DE3 stars that have these capacities for different profiles of codon usage. We might want tunable expression. That's where we would go for with the PBADs, plasmids. And then we might start to get a little bit more elaborate as we get to the scaled production, when we're thinking about autoinduction media and scaling it to a bioreactor production. There's then an extra kind of step beyond that. These are special. I call them specialty strains. So when I think of but they're fantastic in their own rights. And so what I think of for specialty strains is if there's a toxic protein in particular, then you might go for C41s. If you want disulfide bonds, you might reach for the origami strain or the shuffle strain. There's elaborate vector building capacities with Golden Gate cloning or a BioBrick or Bagel Brick cloning. And then, of course, your conditions can get more elaborate too, with scaling up the bioreactors to thousands of liters with defined media and a batch fed system. So all of this is reviewed elegantly in I list one paper here. It's reviewed elegantly in multiple different papers. There's a BL21 variant in particular. BL21 has a filamentation-like phenotype, so that it can form extensive filaments if under the right conditions. And NAB has an NAB Express B strain that's capable of producing protein with that filamentation defect removed, such that they maintain a non-filamentous morphology to them. And that's what I'll be talking about in part today as well. So yeah, because of this diversity, we have these in our freezer boxes. We probably, when we're expressing proteins, if we really like expressing in B strains, we're going to go and we're going to test probably, 5-10 different variants of E. coli. But what if I was to tell you that it might be possible that you could go to your freezer and pull out and test 10,000 different variants at the same time and identify variants that improve the expression of your protein in a very high throughput way. And that's where the magic of the Inscripta Onyx platform comes in. So Inscripta has this solution for CRISPR-based editing, where historically in CRISPR-based editing, you think of in terms of a per well editing, such that each well would have introduced to it a guide RNA or some DNA encoding a guide RNA, and then introduce also the homology repair template to be able to make that modification. But what the Inscripta Onyx platform is fundamentally based on is the introduction of a plasmid that contains the guide RNA, the homology repair template or the donor, and a trackable barcode that doesn't get integrated into the genome and rests on an external plasmid that can be maintained throughout the entire expression process to be able to identify variants of interest in a high throughput method. So that trackable barcode is then in this plasmid. This underlies and is fundamentally based on what's called CREATE plasmid technology that's been well cited. And so we built an automated platform to be able to do this. So to get to the point of developing a method for high throughput generation of variants of E. coli and yeast, we built out an Onyx platform to be able to address anywhere from very specific down to 100 edits, single edits per cell, but 100 different edits in your pool, where you might have very specific knockouts that you might want to make and analyze, very specific promoter insertions. You might, have some idea from literature searches and so forth what to do with in terms of pathway engineering. So you might want to hit certain pathways and be able to perturb certain pathways to see how they help in your protein expression project. And you have the capacity of being able to do protein engineering. So think of like massive site-directed mutagenesis capacities with barcode trackability. What that barcode trackability means is that every variant that you've generated is now accessible through that barcode, which is something that is not accessible with protein engineering on plasmids. And then as you think more broadly, there's all the great stuff that we know about pathways, and then there's all the stuff that we don't know about genomes. And that's where genome discovery and genome engineering comes in. And that's also capable of being accomplished with the same Onyx platform, this automated platform of being able to do genome-wide knockouts and genome-wide promoter insertions that give this capability of discovering all those unknown variables that can improve expression of a protein. So fundamentally, that Inscripta Onyx box is based on a MAD7 CRISPR-based nuclease to do targeted editing. In any given run, we can do from 100 to 10,000 different variants designs within a single tube. That's still a single edit per cell, but each of those edits are then trackable. We have a diverse edit types that can be made from deletions to insertions and substitutions up to 63 base pairs in length. And then we can go gene-centric and do saturation mutagenesis all the way to being able to do genome-wide editing. And then it's left to the creation of the researcher to be able to carry that to higher expression. So the total platform is comes at the entire allowing for the iteration of the design, generate, test, and learn cycle to go much more rapidly. And that all starts at an Inscripta designer. It's an easy to use portal to be able to engineer a microbial genome. It's a lot like, you get to make edits computationally in very high throughput and be able to see the impact of those edits on this elegant designer. You click submit, and 2 to 3 weeks later, you receive a consumables kit that gets loaded to an automated instrument. You press go on the instrument, it's hands off for the next 2 or 3 days, it's working to generate your library containing all of your designs of interest. Once the door opens up, the 2 or 3 days has passed, you take the cell output tube that contains all of those variants that you've generated. And now it's up to the creativity of the researcher to move forward on those with phenotyping. And so you get to pick colonies, look for a screener's workflow, or you can do a selector's workflow. At the end of the day, this all goes back to the sequencer where you can read out those barcodes of the whole genome sequencing, and then you can push that back up to the software that has an automated capability of analyzing the results. And so it's really acting as a kind of collaborator for you for your entire workflow to learn new things about the improvements that can be made with your organism, and then feed those right back in and continuously cycle through to get those type of improvements. So as I briefly mentioned, there's 2 different workflows that we nearly always go through with those edited cells. We take that edited cell pool, and then we either move forward in an isolate-based screening workflow or in a pooled-based selection workflow. Here, in this example, I will mostly be talking about a screeners-based isolate workflow, where then you take that forwards downstream to identification of higher expression or greater activity of the enzyme of interest. And that might come through high throughput mass spec. In my case, I used a lot of plate reading, just simple fluorescence-based plate reading. At the end of the day, I get some type of phenotype readouts from that. And then that goes back to sequencing, as I mentioned, to be able to connect that genotype to the phenotype. And that's really powerful when you think through the capability of being able to generate that relationship to be able to discover new things about your organism and about the expression profile, and even the mechanism of action sometimes on how the protein is actually responding in the cell. So we have now the support for high efficiency editing in B strains in E. coli. So within the last few months, we rolled out the capacity of being able to support editing within B strain. And our supported base strain that's distributable is the Inscripta 1022 B strain. This is derived from NAB Express. And so we see high edit fraction. So the percent edited on the right, I show a table, the percent of the library that was observed when taking just a small aliquot from the cell output tube and processing it by sequencing. We see, nearly 100% of the library is observed on these runs, and the edit fractions are pretty fantastic. I mean, this is, 45% of the aliquot of cells contain edited cells. So the edit fraction itself is quite high. We've tested this in a variety of custom strains as well, besides the NAB Express for D3 type work for where you want that T7 polymerase. There's a T7 Express. For disulfide bonds, we've tested it with shuffle strain and NAB strain as well. And we've also tested it with a number of BL21 strains, both from NAB as well as from Agilent and Novogen. In all cases tested, we see really, great performance with the B strain. And we're really excited to be supporting this capacity now. So I wanted to talk a little bit about 2 stories that we have on improving heterologous protein production with the Onyx platform. The first story is actually in yeast. So we've been talking about B strain. Yeast is another microbe that the Inscripta Onyx platform supports and is capable of doing high throughput editing to be able to identify those type of variants that would improve protein production. So in this case, we had an interest in improving CBH1 production in yeast. So CBH1, cell biohydrolase 1, degrades cellulose and is involved in that pathway. which is really, useful for industrial purposes. So what we did is we generated a base strain with CBH1 variants, wherein we also put a specific signal sequence to allow for its export out of the yeast that we were using. We utilized a SenPK base strain or parental strain that we built this into. And then we expressed it with a 2 promoter and chose a terminator that would also give high expression. So we started with a pretty good expression cassette already integrated into the genome. We had a fluorescence-based assay that worked on, the absorbance at 405 to be able to assay the actual activity of and give a readout on the amount of protein that's available. And then we executed on a screening workflow. And so this execution was done by Eric Abate at Inscripta, where he screened through many different colonies to be able to identify variants, genome variants that improved expression. What kind of libraries did we use, and what kind of libraries are we talking about generating here? So we can go genome-wide with our libraries, or we can do targeted-based approaches. With genome-wide libraries, we can think through applications where we would do genome-wide knockouts, for example, so knocking out each of the individual coding sequences. We can go genome-wide in yeast with terminator insertions that stabilize each of the proteins through that stabilizing element on the 3' UTR. We can also go genome-wide and make modifications to predicted promoters to be able to identify how that might respond to protein production. And then, we call them genome-wide because they're usually spread out, and this works in E. coli and yeast, and we'll get into this with E. coli too, is that we can modify or perturb protease pathways and also, say, glycosylation pathways in yeast to be able to see if those have any impact. And then, so this is the genome-wide, and then we might think of targeted libraries. And this is a different philosophy. This is targeting the actual heterologous sequence that you've inserted into the genome. So as soon as it's in the genome and can be carried around in the genome, that becomes accessible to editing. And so we carried out and executed a number of edits libraries that are targeting the gene specifically itself. So modifying the 2 promoter, for example, by insertions and deletions of transcription factor binding sites, and also swapping around binding sites. We had a library that modified the signal sequence as well as the Kozak sequence, and then a site saturation library where we were investigating novel variants of CBH1 itself. So it got really exciting to be able to automatically generate these types of libraries where the real fun of it comes in the phenotyping that comes off of instrument. We can grab those and do phenotyping and be able to execute on that right away so that we can get from conception to reality very quickly. And what's fascinating is we ran all of these screens with thousands of colonies. Even on those first, what we call the first round, this screening gave a number of different fold improvements over the base strain. So it was, you can see they're hovering around 1, but there's a number of different, in every case of all of these libraries that I just talked through, there's a number of different variants that are hitting at that 1.5 to 2-fold improvement already in that first round of editing. So we're seeing that CBH1 in these strains is already being overexpressed. So what would happen then if we took all of those winners, all of those fantastic hits, recombine them and generate a new library and stacked on top of that library? And that's exactly what Eric Abate did. He took those hits, he generated a new genome-wide knockouts and different expression variants on top of that, as well as the hits library on top of that. What he saw now is that he's starting from, with all of those, rich base strains that are already working well, he's already starting from that 1.5 to 2-fold improvement. And now he's going and getting, greater than 2-fold enrichment, almost up to 3-fold enrichments of that production of CBH1. And this is iterative rounds on the on the instrument that he's executing this on. And so this is done within, months from the conception all the way to now getting greater than 2.5-fold improvement of expression. He then took further those variants that were really outperforming from that second rounds. And already he's starting from a high baseline of about 3-fold improvement on the top hits, and then further does a hits library on top of that, where now he's iterating and he's only taking the best winners from the first library, stacking on top of that, and identifying those that now he's getting greater than 3 folds, greater than 3.5 folds improvement of expression after just, iteratively running 3 rounds on the instrument, looking for variants. And what's fascinating about that is that then you can start pulling out the biology of those individual variants. So in total, he ran 7 libraries on the first round and identified 74 hits. This was within 3 months that he identified that almost 2-fold improvement. He iterated on that and identified improvements that got him above 2-fold improvements in a month and a half. And then he did that third rounds of editing where he got that 3.5-fold improvement, almost 4-fold improvement in yet another month and a half. So in total, he was able to improve his protein production by 4 folds in a little less than 6 months. And when you think through the downstream implications on that on a bioreactor scale, that's an awfully compelling and interesting argument about how much improvement can be made by the host strain modifications. One of the neat aspects of this is that although it took that, nearly 6 months, this was a very hands-off workflow in terms of the actual design and build of these libraries. This was, and oftentimes the Inscripta designer is automated and fun. It's so fun to work with that, it doesn't seem like hardly any time is spent at all with the designer. And then it's that push button go to generate the actual libraries. So ultimately, this is very few FTEs to be able to get this level of improvements in this amount of time. So we wondered, hypothetically, could we apply the same philosophy towards B strain production? And that's where this workflow came in on how we might be able to edit the B strain to improve production. So our target, our first round target was a protein of an enzyme of interest to both therapeutic purposes as well as a bioindustrial enzyme. So the target therapeutic that we had begun looking at was a candidate with this broad use case in acute lymphocytic leukemia or ALL that then also has an additional secondary use case in bioprocessing for the removal of acrylamide from food sources. There's a headwind to greater benefit, though, in this particular therapeutic, and that's that it's immunogenic and so that limits its usage. It's also rapidly degraded, that loses its activity over time, and has a secondary neurotoxicity associated with it, with its secondary enzymatic activity. And so there's a real need for improving the production, improving the enzyme, and yet when overproduced, it's toxic to E. coli by definition from its starvation use case. And so there's a real need for improving also the host to be able to be capable of producing highly active variants. And so I walked through 2 different workflows when I was looking at heterologous expression. First and foremost, I wanted the full use case of the Inscripta Onyx. So I did an insertion of the gene within the genome. That allows for the scalable, stable expression without an antibiotic resistance cassette, and also enables Onyx-based editing with barcoded capacities to be able to track edits. I found that quite powerful. I also wanted to test a plasmid-based expression system through rapid that allows me to be able to do rapid cloning and bring all the tools of molecular biology to bear with these Onyx libraries, and associates back to being able to make rapid advancements on that plasmid-based mutagenesis, albeit not barcoded. So I want to talk about the heterologous gene insertion first. So what I want to walk through is that scale up, the concept of scale up that's happening within just a few months of time. So we had this concept of the protein of interest to look at. So it was a heterologous protein that was engineered to be able to be put onto the genome. We installed it within the Inscripta 1022 genome, and then subsequently also knocked out a native protein that was interfering with the assay. So we've developed an optimal chassis for expression within the first 2 months from concept to getting to that base strain build. So then I did a further rounds of production maximization for the capacity of the protein given its toxicity, utilizing a promoter library that sat in front of the protein. And if you've had an opportunity, you might want to check out the GFP webinar that we've given several months ago that was utilizing a similar type promoter optimization in front of GFP. So here we were optimizing the protein production. Here you can come to bear all of the other gene-specific libraries that we talked about previously, doing promoter-based and RBS screening for improved production, as well as changing any signal sequences or protein tags that are present, and that codon optimization in the first several amino acids can be quite important. And you get, once it's installed within the genome, you get to explore all of the protein function that comes with that mutagenesis. So then the real fun starts as soon as you have that base strain. Then you get to cycle through the and engineer genomes to be able to track the edits, and that uses, again, back to that barcoded capacity of being able to make variants. We can be able to, in high throughput, identify the variants to be able that are showing the improvement. So that's what the workflow looks like. We insert the gene. We do an Onyx engineering run. We take those edited cells and isolate the individual variants of interest. And then we take that towards phenotyping, sequence the outcome from the winners, and then analyze and learn, and then feed right back into the engineering cycle. So the libraries that then we're engineering on, similar to the yeast story, is we built a number of different genome-wide libraries, such as the genome-wide knockouts in the B strain, as well as a genome-wide promoter insertion. So the upregulating the expression of all coding sequences one by one per cell. The cell morphology and folding library is a library that targeted all the cells associated with anything having to do with cell morphology or protein folding by knocking out and dropping in promoters and truncating all of those so that we could look specifically at edits of interest. These would include chaperones, proteases, Z-ring formation, et cetera, et cetera. And then we also have a toxic protein library, and that's scraped from literature reported variants that seem to have improved expression. And these are small insertions, deletions, or single nucleotide variants that just generally were reported. What's neat about this is that, similar to that CBH1 story, with even one round of adatine, we're already seeing a handful of hits that are giving greater than twofold improvements on the activity. So we have an enzymatic activity assay that's fluorescence-based. We did a shallow sampling of only 440 colonies out of the 12,000 different potential variants. So this is a very shallow sampling. And even with the shallow sampling for first round, we got greater than twofold enhancement. So those different hits that we're seeing, we have 6 that are known for stress response genes, 5 for actual cell membrane and stability genes. Surprisingly, 6 genes were of unknown function. And then 5 of the hits were proteases that were knocked out, or chaperones that were upregulated. So we got a really like impressive number of interesting hits, even on the first rounds, that showed improvements of the protein production. But then I transitioned, or I wanted to also, I'm now doing the stacked based workflow, but I wanted to see also if we can improve expression based on the plasmid based expression. And this workflow is a little different. So here we engineered the cells first, and we have the edited cells. I grew those up and made those competent through electroporation. And then I transformed in the plasmids that's compatible with the create architecture that we have, the Onyx architecture that we have. And that transformation is then has carrying along with it the expression cassette of the gene of interest. We isolate that out into individual colonies, phenotype in a similar way on a plate-based reader, and then use sequencing, both Illumina as well as Oxford nanopore sequencing, to identify variants and then analyze and learn from those variants. So directly after editing, directly off instruments, genome-wide knockout library, what I'm showing here is the percent of the library that's observed. So we had 99%, greater than 99% of the libraries observed on the genome-wide knockout, genome-wide promoter, and the cell morphology and protein folding libraries. After transformation with the plasmids, there's some dropouts from, you get a reduction of the diversity that's allowed. And a lot of these are going to be due to the cell death from the actual knockouts, from an actual biological effect. And the other aspect of it is that this is now 79% of your genome is now observable for impacting your expression, which is never before seen. So this is just tremendous to have access to the capability of analyzing biology in this way. So again, we're seeing once again that doing this now with an extremely small shallow sample, again, from the 10,000 different variants that we could have been screening through, I've screened through, just a couple of plates and replicates to be able to see that induction off of the plasmids. And similar to the other two stories is that, again, I'm seeing about a 2-fold enhancement of expression in a couple of different variants. So those ones at the very top, one is surrounding the central metabolism, and another one is associated with sugar or lactose metabolism. And then another interesting hit right at the very top is a gene of unknown function. So similarly, we see that kind of story crystallizing over and over again, that we don't know all of the capabilities of biology. And so we have to access those unknowns. And the best way that we can do that is by engineering and tweaking the genome to be able to perturb it, to be able to see where those novel variants come from. You can see the explosive improvement in expression. I'm really excited to see the stacked variants as I take it through the both the insertion, the genome insertion, as well as the plasmid-based. I think we'll be looking at really, really high expression on some of these proteins. So just in conclusion, the Onyx is now supporting that B-stream functionality to improve protein expression. We have this rapid workflow that we're kind of there, the platform's there to help you at every step along the way, from concept all the way to scale up. And that happens with just a few FTEs in just a couple of months. Even with low depth of screening, we're seeing improvements on the order of 2-fold improvements. We may not, be hitting that jackpot on that very first screen, but it sets us up for success when we do subsequent rounds of editing, taking that agnostic view that we're just looking for optimizing that protein expression. And we also have this plasmid-based expression with a custom plasmid to be able to identify enhancements that are associated with those genome-wide editing libraries, yet still being able to utilize that plasmid-based approach for those users that really like working on plasmids. So just to wrap up, the Inscripta Onyx works with E. coli and Saccharomyces cerevisiae and a number of customer strains that are around those two hosts. We can do single edits per cell, both on the genome itself as well as heterologous sequences. For E. coli, it's 100 to 10,000 different designs per library, single design per cell. And then yeast, it's 100 up to 6,000. And that's insertion, deletion, substitution up to 63 base pairs. And all of this, comes in a consumable, easy to use, load consumable kit in about 3 weeks. So thank you so much for allowing me to present today, and I'm really happy to take any questions. Thank you.
Thank you, Dr. Shepherd, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question here asks, is it possible to perform mutagenesis of a target gene or operon encoded by a single copy BAC for Plasmid in E. coli?
Thanks for that question. That's fantastic. So we are keenly interested in other plasmid-based systems, such as editing on BACs. So I would say reach out to an account executive if you'd like to chat about that. So the plasmid-based system that was used here is a higher copy plasmid-based expression system, but definitely we have ongoing interest as well with BAC-based systems.
Great, thank you. Next question asks, can the same concept be applied to mammalian or human cells or cell lines?
That's a really good question. So conceptually, in theory, it can be applied. The Inscripta Onyx platform is built specifically and currently for E. coli and Saccharomyces cerevisiae work. And so highlighted here is around that improvement of the expression in those hosts. Yeah, thank you.
Great. Next question. He said, thank you for the wonderful presentation. Can the Inscripta system be used in vitro production of siRNA?
To improve siRNA, another really cool question. So here, of course, we were focused on protein production, but I want to say, instinctually, my gut feeling is absolutely, in a way, for in vitro production of siRNA, typically you would use polymerases and so forth, where you might think, how would you apply genome engineering to improve such a system? You could probably focus down or look at improved variants for decreasing RNases or so forth. And these are the type of really good applications for the Inscripta Onyx technology to be able to make high throughput edits to microbial genomes that could improve those type of downstream processes that you might want better purity and so forth from your final enzyme of interest. That's exactly right on to conceptually to how to utilize best the Onyx technology.
Great, thank you. This next question has a few parts, and it asks, how is the plasmid customized? Does customization depend on the protein of interest, or is the customization necessary for co-propagation with the MAD7 nucleus plasmid?
Cool question. So the plasmid itself is customized to be completely compatible with the plasmid-based approach of that barcoding system that's in E. coli. So there we had to be thoughtful just in terms of the origin of replication that we were using, as well as the antibiotic resistance that we were using for it. So once we had that capability of having it propagated alongside the plasmid that carries the barcode with it, that allows us to be able to access that barcode through the Illumina next generation sequencing approaches. Does it propagate along with the MAD7 nuclease? This was designed in such a way that the part of the system that carries the MAD7 nuclease that's fundamental to the Onyx approach. In this case, we actually relieved it of that parts of the process, such that the MAD7 and the repair machinery was absent from these cells. That was part of the plasmid-based choices that we made for expression and open the door to allow us to be able to introduce the plasmid without disrupting or being disrupted by that MAD7 nuclease presence.
Great, thank you. Next question here. You used fluorescence-based approaches here, but how would I utilize this without fluorescence-based phenotyping?
Yeah, so the assays presented here were all, could all be amenable to fluorescence or plate reader based approaches. Historically, we've also used high throughput mass spec for assaying, and that can also be applied within the approaches taken here. So there we would be screening for production of small molecules during the assay. So fluorescence gives you, a pretty straightforward and accessible readout on phenotyping, and of course is amenable to plate-based. And so when you have assays that are capable of, even through tags, looking at protein expression or enzymatic activity through fluorescence, the phenotyping, there are many approaches where the phenotyping can be done in very high throughputs. That being said, even as you saw with shallow sampling, we were seeing some pretty nice hits coming through, and so even a workflow then where it might be a lower throughput workflow, you can still imagine that you're going to see some hits in a low screening based approach. The other approach that I didn't talk much about is a selectors based approach, and a lot of enzymatic activities can be converted into a selectors based approach with some genome engineering involved, and so that's something to be thoughtful as well about. That really opens the capacity of exploring all of the variants in a selectors workflow.
Great. Next question, is this webinar or slides going to be available for download?
Yes, I believe that the webinar will be available for downloads fairly soon, or watching online again.
Awesome. Next question here. How could I get some of your yeast libraries or clones for verification purposes?
Interesting question. So for now, if of interest for reaching out for any project ideas that you might have, I might point you towards contacting an account representative, or an account representative might be in touch. But that can help to identify if there's an interest for projects of specific interest or purpose, and then also just getting in touch with us if you have some types of those questions. That's great.
Great. We have time for a few more questions here, so we'll continue on. Are the improvements you're seeing with your edits generally helping protein expression, or are they specific to the protein?
This is a good question. We have an interest in exploration of this question as well, and I think the plasmid-based approach becomes amenable to be able to test in high throughput. So the answer here is going to be a little murky because I only showed data for one particular protein or one particular enzyme in E. coli. But now with that opportunity to be able to screen in high throughput genome engineering, that would allow us to be able to observe with multiple different proteins if it's a general, if the modifications have general utility of plasmids across the board or proteins across the board, or focus down on sometimes that's going to be the case, and other times as you're screening through, what we would presumably find is that other edits are specific to an individual protein and the activity of the individual protein. So I think the Inscripta Onyx platform gives us a unique capacity to be able to discover if it's a general phenomenon versus a specific phenomenon on proteins, and really it takes that iterative workflow to be able to discover specific host variants that can really, hit the ball out of the park in terms of expression of any given specific protein is the way that I look at it.
Great. Well, it looks like we have time for one more question, so we'll wrap up here. What type of improvement would you expect had you screened all the edits? And why did you only screen just a few hundred in the E. coli?
Yeah, we've often seen this is a good question. So when proteins are expressed, when you create variants with the Inscripta Onyx platform, because of the high throughput nature of it, you're routinely generating libraries of greater than 10,000 different variants that might have a specific impact on the expression profile. And so one philosophy might have that you would like to be able to see what every variant in that pool, all 10,000, what the change in capacity is. But really what we've found over and over again is that time is better spent by doing shallow sampling and with a fairly low screening, and then stacking on top of your hits. And so we saw that with like the CBH1 yeast story, that we for sure saw like this, fourfold improvement once we're stacking on top of with shallow sampling. And we want to port that over and do that with the B strain as well, because we think that the same philosophy, we've seen that with lysine production, small molecule production, and we've seen that with tolerance to growth inhibitors, where shallow screening, several colonies, and then stacking on top of those adds richness to the library well beyond what we would get if we spent all of the same amount of time or all of our time trying to screen through all 10,000 different variants.
Great. And we got one more question that just popped into the chat that we have time to answer real quick. Will you introduce K-strain engineering?
Fantastic question. So K-strain engineering is a sweet spot and definitely supported. We've been doing that approach. We've been working with MG1655. We're definitely ready with K-strain engineering as much as B-strain engineering.
Fantastic. Well, thank you again, Dr. Shepherd, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.